welcome to another lecture. This lecture will be about connective tissue. I hope you have a nice day, so let's start. First, we will look at the definition of connective tissue, which is very important. Connective tissue is generally that we have cells that are embedded in extracellular matrix. So we have some cells and some extracellular matrix, which is produced by these cells. So cells are very important, and also extracellular matrix is very important. And we have three basic types of connective tissue. We have proper connective tissue, a cartilage, and a bone. So let's start with the description of the cells. Even though they do not form the main mass, but they are essential for the production of mass. We divide them into resident or fixed ones. Yeah, so we have resident cells and we have migratory or wandering cells here. These resident fixed ones originate in actual tissue, and their main task is to produce extracellular matrix. Yeah, so here we have some kind of undifferentiated mesenchymal cells, which differentiate into several types in each connective tissue. Then we have migratory cells that come from the bone marrow. So they do not originate in the actual tissue, but they come from the bone marrow. They stay for a while in that tissue and they go away right after, or maybe stay there, but they do not originate in the actual tissue. And what does that mean is that their precursors are white blood cells. So we have some kind of hematopoietic stem cells in the bone marrow. Yes, stem cells in the bone marrow create several types of cells, and a monocyte, which is a white blood cell, then differentiate into, for example, free macrophages or osteoclasts, which are in the bone. And these are the types of migratory cells. Or for example, these stem cells differentiate into B lymphocytes, which then transform themselves into plasma cells, which produce immunoglobulins. You already know plasma cells. And plasma cells are also migratory cells of the connective tissue. Yeah, and for example, also mast cells. So migratory cells are osteoclasts and free macrophages, which differentiate from the monocyte right here. Monocyte. Plasma cells are another type of migratory cells, and mast cells. Yeah, so these are very important. So remember, they spend only part of their life cycle in the tissue. And really the main fixed cells of the proper connective tissue are fibroblasts. So main fixed cells of proper connective tissue are fibroblasts. And then we have, for example, a cartilage, and main fixed cells there in cartilage are chondrocytes. Yeah? Osteocytes are in the bone. Adipocytes are in the adipose tissue. So main types are fibroblasts, chondrocytes, osteocytes, and adipocytes. These are the main types. Also, as for the intercellular or extracellular matrix, this matrix is divided into two main components, and that is a fibrillar component and amorphous component. The fibrillar component consists of the protein collagen. Collagen is the main protein of fibrillar component. And this collagen is in structures called fibers. Only some types of we have many types of collagen, and only some types of collagen create fibers. Yeah, and we have also elastic fibers. Yeah, so don't forget about them. So collagen fibers and elastic fibers are the main structures of fibrillar component. The amorphous component is less observable. It's formed by molecules, so we really cannot see amorphous component. We cannot see molecules in the light microscopy, so they are less observable. There are three types of macromolecules, and these are the GLYCOSAMINOGLYCANS, proteoglycans, and the structural glycoproteins. These are the three main types. For example, glycosaminoglycans, we know many of them, and only hyaluronic acid, which you sometimes put on your skin and is very healthy, but hyaluronic acid is also in the tissue, and this acid can occur alone. Yes, so you have free hyaluronic acid in a proper connective tissue. But other macromolecules, other glycosaminoglycans, which we are going to discuss later, needs to be bound to a protein. So other glycosaminoglycans are bounded to some protein carrier. Yeah, so we have these proteoglycans. Proteoglycans are bigger structures, and proteoglycans are glycosaminoglycans that are bounded to some protein core or some protein carrier. So these are proteoglycans. And the last group are structural glycoproteins. And these are just the proteins, big proteins, which are linked with saccharide chains. Yeah? So there is this difference. In structural glycoproteins, so protein here is the main component. Protein here is the main component. On the other hand, proteoglycans, which are these glycosaminoglycans linked to a protein core, glycosylated structures or saccharides are the main component here. Yeah, so the main component is represented by the second part of these words. So, there is a picture of some connective tissue, proper connective tissue. It's very normal. And so we have here, we can see some cells over here. So these are the nuclei of some cells, so many cells. And between these cells, we can see some amorphous component. And sometimes it's more homogeneous, like right here. Sometimes it's more homogeneous. And sometimes we can see these fibers right here. So sometimes we can see like some structures like these, like the fibers between cells. And also connective tissue contains a lot of vessels right here. Here's the vessel, right here. So proper connective tissue is well vascularized. These arrows are pointing to the vessels here. As you remember, epithelial tissue has strong regenerative ability. But for the connective tissue, the situation is somehow different, because not all types of connective tissue can regenerate. For example, cartilage is not able to regenerate at all, or only in very, very small amount. But for example, proper connective tissue like this is able to regenerate. So it differs. It's very different. Oops. So next. And let's go for the synthesis and secretion of collagen. Collagen is the most important or most common protein in the human body, of course. And it's a secretory protein, so it's formed on the ribosomes, which are attached to the endoplasmic reticulum. And really, so we have some messenger RNA that produce, in the process of translation, a polypeptide chain is produced. Yeah. And there are some post-translational modifications going on that start on endoplasmic reticulum itself. So the first post-translation modification is hydroxylation. Hydroxylation, right here. So what did I want to say is that only some proteins, only a few proteins undergo hydroxylation. Yeah, only a few proteins undergo hydroxylation. Specifically, proline and lysine, which are amino acids, undergo hydroxylation, and we need vitamin C in order to do so. So we need vitamin C to hydroxylate a collagen. That is very important because when there are no hydroxide groups on the collagen, it's not able to function properly. So vitamin C is very important for that. And next post-translational modification is glycosylation. Some molecules are more and some are less glycosylated. All this is already happening on the endoplasmic reticulum. So also hydroxylation and glycosylations are happening in the endoplasmic reticulum. And as we can see right here, a collagen polymerizes into a triple helix. So here we have a triple helix right here. So we have 3 chains that polymerize into triple helix. And this triple helix, the ends of the triple helix are marked with short peptides called registration peptides. So we have 3 registration peptides right here on one end, and we have another 3 registration peptides on the other hand, and they prevent premature polymerization. So we have this triple helix that has registration peptides on one end and on the other end, and they prevent polymerization. Because really, what's happening here and why do you want to prevent polymerization is that, like, you don't want collagen to polymerize in the fibroblast, because fibroblast will die in that case, because collagen is very big structure. It's even bigger sometimes than the whole fibroblast. So you need to prevent polymerization in the fibroblast. So collagen that is not able to polymerize, collagen that has these registration peptides on each end is called procollagen. It's called procollagen. In the Golgi complex, it receives a membrane, so it's not stored in the cell, but it's immediately excreted from the cell in the form of vesicles. So fibroblast isn't storing these collagen proteins, these procollagen proteins, because it's very dangerous to store them. Maybe they start to polymerize in some time, so it just secrete it in the form of vesicles outside from the cell. And this type of secretion is called constitutive secretion, that you are not storing your product in the cytoplasm. So what's happening right here? The collagen reaches the cell surface. It reaches the cell surface. And when it's still in a contact with the cell membrane, the registration peptides are cleaved. Yeah, so it's in the procollagen is in the vesicles, and when it's in contact with the cell membrane, the membrane of a fibroblast, these registration peptides are cleaved. And why is that so? It's because the membrane of a fibroblast contains peptidase, which cleaves the registration peptides out. Yes, so membrane of a fibroblast have registration, it has got peptidase. It's an enzyme that cleaves registration peptides from the molecule of procollagen. Yeah? So that's what is happening. And when we have a triple helix of collagen that hasn't got these registration peptides, it's called tropocollagen. Or sometimes only collagen molecule. Yeah, so when we have this triple helix that is shortened a bit, it doesn't contain these registration peptides, we call it a tropocollagen. And this tropocollagen is able to polymerize. So the prefix, the tropo, is no longer used, so it's not now called only a collagen molecule. And when we say a collagen molecule, we really think of tropocollagen, the collagen that is able to polymerize and doesn't contain registration peptides on the ends of it. So, tropocollagen is released from the cell. Yeah, in the form of vesicles, it's released and it's able to polymerize. And it's doing so. So it does this polymerization. Polymerization occurs near the cell, in such cells in vaginations. For example, here, 10 and 11, they're here, can tropocollagen polymerize right here. And the first products of polymerization are protofibrils. And remember this, that all types of collagen are able to polymerize to this stage. So every collagen that's out there can create protofibrils. So once again, we have messenger RNA that creates collagen chains that undergo hydroxylation and glycosylation. This process starts on endoplasmic reticulum, but main glycosylation occurs in Golgi complex. Yeah, they form a structure called procollagen, which has registration peptides on ends. And when they are cleaved, because of peptidase in the membrane of fibroblast, they create something called tropocollagen, or just collagen molecule, which doesn't contain registration peptides and can be polymerized in that case. And with polymerization of tropocollagen molecules, we create protofibrils. And first we are going to describe tropocollagen, or only collagen. The length is about 280 nanometers, so this is the length of this molecule. The thickness is about 1.5 nanometer. Yeah, so it's just a little about the observable limit of the electron microscope. And really what we can see right here, the chains of tropocollagen. So remember, it's triple helix, and these chains can be different. Yeah, can be different, or they can be the same. Yeah, but most of the time they are different. So remember, it's a triple helix, doesn't contain registration peptides. It's about 280 nanometers in length, and it's about 1.5 nanometer thick. And these tropocollagen molecules can polymerize. So here we have one tropocollagen. And when they polymerize in this horizontal sequence, so we have tropocollagen, 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 like this, they create a structure called protofibril. So this right here is protofibril, right here. So really, protofibril is like horizontally attached tropocollagen molecules. And these protofibrils are able to aggregate. So they can aggregate. So they are side by side. So there is another protofibril right here and another one. Yeah, so they aggregate side by side. And they create a structure called a fibril. So they create a fibril right here. So fibril is just created by protofibrils, which aggregate side by side vertically like this. Or they create a 3D structure. Actually, they create a round structure. So really, fibril is composed of protofibrils placed in parallel in such a round shape, and a fibril is formed, which is from like 20 to 75 nanometer thick. And what's interesting is that those protofibrils are not added randomly. Yeah, these protofibrils, as you can see here, they are not added randomly. So there's some kind of structure they are forming. It's not a randomly just the organized structure. There is like certain shift in the tropocollagen molecules, and there is this period that is about 64 nanometers big period. And really what we can see on a fibril in electron microscopy, we can see some dense and some electron-lucent strips through fibril. As you can see right here, this is electron-dense strip, and here is electron-lucent strips. And really that's the question, why some parts are more and some are less dense. And that can be explained by the bonding of heavy metals here. Because metals bind to sites where two tropocollagen molecules bind together. So these loose sites right here. There is a connection between one tropocollagen and the other tropocollagen molecule. And the heavy metals bind right here in this place. And these places are called lacunar sites or lacunar places. So lacunar regions are therefore electron-dense regions. And here in the lacunar places we will find bonds between tropocollagen molecules. Here. So we can see it's very regular period. It's 64 nanometers. Now they are telling us that it's 67 probably. I think it really doesn't matter that much if it's 64 or 67. But what we can see in electron microscope right here, it's the next picture, we can see this stripping right here. And it will be used by to measure size, really. It will be used to measure size because we are in intercellular mass or in extracellular matrix, and we do not have ribosomes here because we are not in the cytoplasm of a cell. So of course we can only help ourselves with collagen fibrils that are here, and so we can see the period that is 64 nanometers. So this right here, this line is about 64 nanometer wide. Yeah, so that's really the period right here. So one lucent strip and one dark strip is 64 nanometers wide. So we can use it as a standard measurement in electron microscope. And really what's happening next is that fibrils tend to aggregate more or further aggregate. Really, they're creating structures called fibers. So we have protofibrils which aggregate into fibrils, and these collagen fibrils aggregate into collagen fibers. And we can see these fibers in a light microscope. So we can see these fibers in light microscope. We are not able to see fibrils, collagen fibrils, in light microscope. We can only see them in electron microscope. Yeah, so these are structures that we can see in electron microscope. Fibers we can see in light microscope. And sometimes these collagen fibers aggregate into the bundle of collagen fibers. And only collagen type 1 can do it. So collagen type 1 can aggregate into bigger structures like bundles, and we can see these bundles by eye. Yeah, so we can see, this is not a very good picture of an eye, but you can like imagine. So this is very important. So when I go back, once again, I will explain this picture because it's very, very important. So these structures right here, these are the fibrils. So we cannot see actually the protofibrils here. These structures are fibrils. So this is fibril, 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 fibril. There are many of them, fibrils, and you can see some fibrils are going another direction right here. And these fibrils, when they aggregate, they create a fiber. So this whole thing right here is a fiber, collagen fiber, and also this structure right here is a fiber. So there are two fibers going in some kind of directions here. That's what we can see right here. This is a picture from atomic force microscope right here, atomic force microscope. So we can see 3D structures, and we can see also the stripping on these structures right here. Yeah, so these are the collagen fibrils right here, and we can see the stripping also. And these are the fibrils right here. So this is one fibril, the second fibril right here, and we can see the stripping through the fibril. So once again, these electron dense structures right here are places where tropocollagen molecules are connected together, and we have heavy metals here in this bonding. So that's why it's visualized like electron dense structure in electron microscope. Because in the process of contrasting, because you want to prepare samples for electron microscope, and in this process you put heavy metals on the samples. So that's why they are heavy metals there in this loose bonding. So once again, in general, we have many types of collagen. And we divide collagen into two basic groups, and that is a fibrillar collagens and non-fibrillar collagens. So most collagens form fibrils, like here, in addition to type 4 collagen, which does not form fibrils. So the most important are the first ones, like collagen 1, 2, 3, which form fibrils. And then we have this collagen 4, which doesn't form them. This collagen 4 is located in basal lamina, in lamina densa. For example, there is a collagen type 7, which you already know, that forms anchoring fibrils in basement membrane, and also a collagen type 6, which is in external lamina of muscle tissue or in cartilage. But these are the most important: type 1, 2, 3, and the 4, which is in basal lamina. Really, the type 1 collagen is the only one capable of forming bundles of fibers. You can see there are these 4 basic types, and fibers are formed in collagen type 1 and in collagen type 3. So in collagen type 1, the fibers are called collagen fibers. So when we say collagen fibers, we basically mean fibers which are made of collagen type 1. Yeah? So, and we can find them in proper connective tissue, in cartilage, fibrocartilage, and in bone. Yeah, so that's the collagen type 1. And these collagen fibers, these collagen fibers have the strongest fibrils that are about 75 nanometers thick. So the biggest fibrils are in the collagen type 1. Then we have type 2 collagen. Type 2 collagen, which has thinner fibrils, only 20 nanometers thick fibrils. But this type of collagen, the collagen type 2, is unable to aggregate into fibers. So we don't have fibers from collagen type 2. And this collagen is absolutely typical for cartilage. That is very important. It's absolutely typical for cartilage. And then we have a collagen type 3, which forms something called reticular fibers. So when we say reticular fibers, we mean fibers made of collagen type 3. And these fibers are located in proper connective tissue. Yeah? So really, we have 2 types of fibers. We have collagen fibers that are made of collagen type 1, and we have reticular fibers, which are made of collagen type 3. They really differ in occurrence and thickness. Yeah, these collagen fibers right here. And then we have collagen type 4, which is located in basal and also superficial laminae. So we already know basal lamina, which is under polarized epithelial cells under the basal part, under epithelium. But really also some cells have also this basal lamina around themselves, but they are not polarized, so we cannot call it basal lamina because they don't have base actually. So we call it a superficial lamina, but it's really the same as basal lamina. Yeah, superficial lamina is really the same, but it's all around the cell. So that's why it's called superficial lamina. And this type 4 collagen does not form fibrils, and there are no fibers. So also no fibrils and no fibers. That's very important. So we only see like the mesh-like structure made of tropocollagen and maybe protofibrils, some protofibrils, but no fibrils at all and no fibers. Because really like what I wanted to say. Yeah, this is the picture from electron microscope, and here we can see the fibrils right here, these structures, these lines here. They are striated, or you can see the stripping with this period, 64 nanometers period right here. So these are the fibrils, and they make up the whole collagen fiber. So this is a collagen type 1 here in this picture, and they make up a fiber. This fiber can be seen also in light microscope. So we can see fiber in light microscope, but we are not able to see these fibrils, of course, because as we said already, they are 75 nanometers thick. So we are not able to see them in light microscope, only fibers, only fibers. Yeah, and collagen fibers can be stained also with hematoxylin eosin, and because collagen is a protein that is basic due to very large amount of hydroxylation, so it's a basic protein, so it's stained with eosin, which is an acid. So in hematoxylin eosin staining, collagen is eosinophilic. Yeah, we can see it right here. Here we can see very thin collagen fibers which are stained with eosin, but also here we can see thick collagen fibers. These structures right here, these are thick collagen fibers stained with eosin. So they can be stained with acidic colors like eosin, but also we have aniline blue. So aniline blue is also acid, and it stains collagen, aniline blue. And we can use this blue trichrome, or sometimes called Mason's trichrome. And this trichrome can be blue, like in this picture, but it can also be green, yeah, and yellow. So really, there are many stainings here that can stain collagen fibers. And this is a picture from a cartilage. Here we can see a very big cell. This is a chondrocyte. And we already know that type 2 collagen doesn't form fibers. So we can't see fibers in light microscopy, or we only can see some homogeneous structure in light microscopy in cartilage. But in electron microscopy, we can see fibrils that are formed from collagen type 2. So these dense structures, the dense circles right here in extracellular matrix, these are the fibrils right here. And you can see we had to use very big magnification to see them, because these fibrils are very thin. Collagen type 2 fibrils are very thin. They're about 20 nanometers thin, so they're very small compared to collagen type 2 or 3 fibrils. So really what we can see in the electron microscope, we can see the fibrils, but in light microscope, it looks like the fibrillar component wasn't even there in light microscope, only homogeneous matrix. And you will see that in the next lecture about cartilage. The next is collagen type 3. Collagen type 3 create reticular fibers, but what we can see in electron microscope here is some kind of cell, yeah, some kind of epithelial cell with a basal lamina. Yes, so here is the basal lamina, and you know right under basal lamina we have reticular lamina, which together with basal lamina create something called basement membrane, and we can really see reticular fibers right here. but fibers are really these whole structures right here. These are the fibers, and these electron dense circles right here, these are the fibrils, reticular fibrils. Yeah, so fibrils right here. Yeah, reticular fibrils. These fibrils together create the bigger structures called fibers, reticular fibers. Really, these reticular fibers have typical places of their occurrence. They are under epithelium. They form this reticular lamina, and really these fibrils are thinner and they are irregular. You can see it right here. We can see longitudinal sections and cross sections, so they are really arranged irregular. And these collagen molecules, collagen type 3 molecules, are quite glycosylated, so they do not connect tightly, and we can see spaces between these collagen fibrils right here, and that's because they are heavily glycosylated. These reticular fibers are heavily glycosylated. They create also these the stroma of some glands, for example in the spleen and also in lymph nodes. This collagen type 3 create these reticular fibers stroma, and we can see it with a reaction called PAS reaction, periodic acid Schiff reaction. And we can visualize them with this reaction because they are heavily glycosylated. But PAS isn't used here. What is used here in this picture is impregnation. So we see these dark lines, and these are really the reticular fibers here. This is a picture from light microscope, so we can't see fibrils. What we are able to see are fibers. So these are the fibers right here. So the situation is kind of different from the previous picture. They're creating this stroma, fibers. And between these cells, we have between these fibers, we have cells which create the parenchyme of the organ. So don't worry about that right now. Just focus on these fibers right here, dark fibers which are impregnated. So they create a supporting structure of the organ. Not in all organs, but some of them, and you are going to learn about them. So they created these networks, the spatial networks. That actually means reticular in Latin, like networks. And really in this picture from electron microscope, we can see the difference between a collagen fiber right here and reticular fiber right here. So here we have, this is a collagen fiber, so collagen type 1 right here, and right next to it we can see a collagen type 3, collagen type 3 or reticular fiber right here. And these are the cross sections, both are cross sections. And in collagen fiber, we can see that collagen fiber has regularly arranged fibrils. So these fibrils right here, they are thicker than in collagen type 3, and really they are regularly arranged, closely packed together, creating this collagen type 1 fiber. In collagen type 3, these fibrils, which create the fiber, are smaller, they are thinner, and they are not so much packed together. There is a space in between. Yeah, we already said that they are heavily glycosylated, so there is space in between these fibrils, and they are not so regularly arranged. We can see that these are the cross sections of fibrils, and here we have longitudinal sections, so they are creating this meshwork. So they are not so regularly arranged like in collagen type 1. So these are the main differences. But it's really, sometimes it's very hard to tell which is a collagen type 3 and collagen type 1. In this case, we can compare them, so it's easy to say, but sometimes it's really hard to say that these are the collagen type 3 fibers. So don't worry about that much. But you should know they are located under epithelial cells. So they are creating this lamina, reticular lamina, so you should know that. And also that they sometimes create this stroma in the organs, such as spleen and lymph node, for example. Yeah, so, okay, I was correct. And also collagen type 4, once again, and 7. This is once again epithelial cells with a basal lamina under epithelial cell, and collagen type 4, which does not form fibrils or fibers, no fibrils or fibers, is located in lamina densa. So lamina densa or dense lamina right here. So here we can find collagen type 4 fibers, making those tropocollagen molecules or protofibrils, the maximum. So it's made of tropocollagen and protofibrils in lamina densa. That's very important. And also type 7 collagen is making those anchoring fibrils, which connect collagen type 1 to the basement membrane. So you should also know collagen type 7, that is also in the basement membrane. So 4 and 7 are in a basement membrane. And here is a picture from the muscle tissue. These are very typical structures. What we can see here, these are typical structures for the muscle tissue. So here we can see a muscle, and there is a surface of a muscle tissue right here. And here we have also a superficial lamina right here. So it's the structure that is same to basal lamina, but it's around the cell, the muscle cells or the muscle fibers, and it's called superficial lamina. So it's really the same structure as basal lamina right here. This superficial lamina surrounds muscle cells or muscle fibers, and also adipocytes, which are fat cells. So it's very typical for them that they are surrounded also with this like basal lamina. Okay, so that was for the collagen fibers, but we also have some other fibers, and these are the elastic fibers also here in a proper connective tissue. Elastic fibers have two major components. Here we can see elastic core in the middle and microfibrils that surround this elastic core. But first, what's happening in the course of production, these elastic fibers, microfibrils are formed from the fibrillin protein. So first we can see the formation of microfibrils from the fibrillin, this is the protein, and this protein polymerizes into like 10 nanometers thick microfibrils. So these microfibrils are like 10 nanometers thick, so we can't see them in light microscope, only in electron microscope, microfibrils. And these microfibrils form a skeleton, yeah, supporting structure of the elastic fiber. And there is this protein called elastin that is in a form of proelastin deposited in the center. Yeah, so first we have microfibrils around, and then elastin core is formed in the center, and it's creating some kind of gummy mass or gel-like structure in the center, and elastin is formed from proelastin. And as you remember, we also had this collagen. We now have proelastin, which is forming elastic core. So the pre suffix is for something that was before, of course. So we have also proelastin. There are many types also of elastic fibers, because these elastic fibers mature in time, but you don't have to worry about that. They are usually in proper connective tissue, but we can find them also in elastic ligaments, elastic connective tissue. Here we can see some pictures, maybe, which are next. Here are some pictures from electron microscope and also from light microscope of elastic fibers. And in electron microscope, we can see these lines right here, thin electron dense lines right here. And these are the microfibrils right here, microfibrils. So there is a one component which are located more like peripherally around the elastin core, which is in the middle. And this electron dense material is elastin core right here. So two components make the whole elastin fiber. Yeah. So elastic fibers can be stained with hematoxylin eosine. They are stained with eosine, but not so well. They are more light. They are lighter eosinophilic than collagen fibers. Here we can see it. This is a picture from light microscopy, and these are the elastin fibers right here. But you can see they are pink, but not so pink as collagen fibers. So that's typical for them. But we can stain them with special staining. This is a picture from light microscopy. And you can see here we have elastic fibers which are black. Here in this picture. They are black. They are stained with a color or staining called resorcinol. I think that's the right name for the staining, probably resorcinol fuchsine. This is a picture from the vascular wall. So especially vascular walls have many elastic fibers because they need to be elastic. They need to dilate and go back to the normal position. So they need to be elastic. So they have a very large amount of elastic fibers. Here we can see them. These lines, black lines, are elastic fibers. And sometimes these elastic fibers, especially in the vessels, they form membranes, whole membranes. So they are not separated, and they create this mesh-like structure in the walls of vessels, and these are called fenestrated membranes. Yeah, or in Latin, membrane fenestrating. Yes, something like this. Fenestrated membranes. That's in Latin. I wrote that down. So these are very typical for vessels right here. So really just remember, elastic fibers can stretch, they can shrink, they're very elastic, that's why they call it elastic fibers, and they are made of two components, the microfibrils, which are around, which are creating this skeleton, and elastic core, which is this amorphous component in the middle, which is made of protein called proelastin protein. And we can find them in proper connective tissue and also in a very large amount in the vessels, in the wall of vessels, and in elastic components, or elastic ligaments, I should say, elastic ligaments. So, and here we have glycosaminoglycans. So let's talk about amorphous component of proper connective tissue. So once again, glycosaminoglycans are the first structures or the first molecules that can be found in amorphous component, and you can see there are many of them. We have something called chondroitin sulfate, because these guys right here, these four guys, are sulfatated. So that we have dermatan sulfate, heparan sulfate, and keratin sulfate, and these glycosaminoglycans are located in for example, the dermatan sulfate is also, of course, in the skin, but we can find it in tendon, aortic adventitia, which is a one layer of aorta, or chondroitin sulfate, of course, can be found in cartilage because they interact with the collagen. So, for example, this chondroitin sulfate, chondroitin sulfate has very high ability to interact with a collagen that is located in the cartilage. So collagen type 2 is in the cartilage, and this chondroitin sulfate really likes this collagen type 2. So that's why we can find it in here. And also we have this hyaluronic acid that I mentioned before, which can be alone in the tissue. Doesn't have, the hyaluronic acid doesn't have to bind to protein. These four guys here need some protein carrier. They need some protein to be there. They connect to proteins, but hyaluronic acid can be on its own. Yeah, and we can find very large amount of hyaluronic acid in umbilical cord, in synovial fluid, which is a fluid in the joints. Yes, synovial fluid, it's in the joints. Humor vitreous, which is in the eye, and also we can find it in cartilage. Yeah, also in cartilage. And you should know these glycosaminoglycans, and you should know where you can find them and what type of collagen they interact with. Yeah, so for chondroitin sulfate, it's very clear that it's in the cartilage, for example, chondroitin, and dermatan sulfate is in the skin. Yeah, so you should know probably some of them, not all of them, but some of them, the location. Yeah, so, and you should know this interaction with collagen. So when you know that the dermatan sulfate interacts with collagen type 1, so you just know where you can find it. You can find dermatan sulfate in the structures where a collagen type 1 is located. Yeah, so it's not just skin, tendon, aortic adventitia. There are many other structures where you can find dermatan sulfate, for example. Just remember these four needs a protein carrier, and hyaluronic acid isn't sulfatated and can be on its own in amorphous component. Yeah. Next we have proteoglycans. This is what proteoglycans really look like. They have different names, and for example, aggregan, biglycan, perlecan, versican, lumican, they end with can, and because they are proteoglycans. And really these molecules look like interdental brushes, I think so. Yeah, so we have some kind of protein carrier with glycosaminoglycans here attached to the protein carrier right here. And they are forming proteoglycans. So the main structure in this molecule is are glycans. Yeah, so we have more saccharides than proteins in this structure. You should know the names of some of them at least. Yeah, aggregan and biglycan is very important. And the last molecules are structural glycoproteins, and they have very different names once again, but it's not that hard to tell in which tissue you can find them, because once again we have fibronectin, which is typical for proper connective tissue, fibronectin. Chondronectin is typical for cartilage. Osteonectin is typical for the bone. And these structural glycoproteins are very important because they interconnect many parts in extracellular matrix. For example, you have a picture of fibronectin here, and you can see that fibronectin dimer has different sites for binding. Fibronectin has heparan sulfate binding sites, so it binds to heparan sulfate, which is a glycosaminoglycan, and they have also cell binding sites. So fibronectin is able to bind to some cells, for example fibroblasts in a proper connective tissue. Yeah, and we also have this collagen type 4 binding site, so they also are able to bind in the basement membranes or somewhere where collagen type 4 is located. So really they interconnect. Yeah. So when there are more and more structural glycoproteins in the tissue, the more interconnections are made in this tissue. Yeah, so just remember they're important for interconnecting all stuff in the matrix. Yeah, and also cells to the matrix. Yeah, so that's why they're called structural glycoproteins. They have very, very difficult structure. Like for example here, laminin, very difficult structure, many binding sites to many things in the cell. Yeah, once again, cell binding site, proteoglycan binding site, collagen binding site. So they create these networks between everything. For example, the laminin and tenascins are in the basement membrane. Yeah. So now let's talk exclusively about proper connective tissue. Just remember, connective tissue can be divided into proper connective tissue, the cartilage and the bone. And we are going to talk about proper connective tissue right now, and cartilage and bone will be in the next lecture. So once again, connective tissue is made of cells and extracellular matrix. And cells in proper connective tissue can be fibroblasts, fibrocytes, myofibroblasts, reticular cells, adipocytes, and also cells which are not included here, but they are called mesenchymal cells. But mesenchymal cells can be found only in embryo. Mesenchymal cells only in embryo, because that's really these cells, mesenchymal cells, are really undifferentiated. So in normal human body, in normal proper connective tissue in human body, we can find fibroblasts which produce collagen. Yeah, and they can produce collagen type 1 and also collagen type 3. These cells are very active. We're going to describe them. Inactive state of a fibroblast is called fibrocyte. So these cells are the same as fibroblasts, but they are not so active. The nucleus of these cells is darker, you're going to see, and they don't produce as much as the fibroblasts. Then we have myofibroblasts, which contain some structures that are also in the muscle tissue, so they are able to contract. So that's why they called myo. This is a prefix for the muscle, myofibroblasts. They contract themselves. And then we have reticular cells, which are very special cells in terms of production, because these cells are able to produce reticular fibers, and reticular fibers, that's collagen type 3, as you already know, so they can only produce collagen type 3. Yeah, remember, fibroblasts can produce also type 1 collagen and type 3. Reticular cells are specialized and they can only produce reticular fibers. And then we have adipocytes, which are the fat cells that store fat, and these adipocytes are very special cells. We're going to describe them. So these were the resident cells or fixed cells, which stay in the proper connective tissue. And then we have these migratory cells, which are derived from the bone marrow. They origin in the bone marrow. And we have these macrophages, which are made of monocytes, mast cells, plasma cells, which are effector cells of B lymphocytes. Yeah, plasma cells produce immunoglobulins, and also some leukocytes and lymphocytes can be found in the connective tissue. Normal free granulocytes and lymphocytes, you can find almost everything in proper connective tissue. And then we have this extracellular matrix that is around the cells, which are in the connective tissue, and we can find fibers in light microscopy, fibrils in electron microscopy, and also in proper connective tissue we can find also collagen type 1 and collagen type 3 that form fibers, collagen fibers and reticular fibers, but also these other types of collagen like collagen type 4 in the basal lamina and collagen type 7 also in the basement membrane, and collagen type 6 and other structures, and my pen fell down. So I'm overly then So these are the fibrils and fibers, and this is the fibril component of extracellular matrix. And then we have amorphous component in extracellular matrix, a gelatinous component, once again made of glycosaminoglycans like hyaluronic acid or dermatan sulfate or other structures that can be molecules that can be found there. So and fibronectin as a structural glycoprotein is important because it connects everything together, and fibronectin is very typical for the proper connective tissue because it's a fibronectin, and we have fibroblasts and other things there. So let's go for the cells right here. As I mentioned before, we have mesenchymal cells, but you are not able to see mesenchymal cells probably in our slides. Maybe we have some new slides right now from the embryo, so probably there you would be able to see mesenchymal cells, but what you are able to see almost on every slide is a fibroblast, because connective tissue, proper connective tissue is everywhere. So you're able to see fibroblasts right here. Yeah, so these are the fibroblasts, and you can see the nucleus is light basophilic nucleus, which is very active nucleus. These fibroblasts have basophilic cytoplasm because they need to have granular endoplasmic reticulum. And you know that fibroblasts produce collagen, so a collagen is a protein, so they need ribosomes, and they need granular endoplasmic reticulum because these collagen proteins, are excreted from the cell. So that's why they have basophilic cytoplasm. I know it looks like kind of pinky in this picture, but really you need to think about it before you said that this cytoplasm is eosinophilic, because it's not. It contains a lot of ribosomes, so it's basophilic. The cytoplasm is basophilic. And they are more like spindle-shaped, sometimes star-shaped, with oval nucleus. So these are the fibroblasts. And around we can see some collagen fibers right here, very thin collagen fibers. And inactive fibroblasts are called fibrocytes. So these are really inactive cells. You can see the nucleus right here is darker than the nucleus right here from a fibroblast. So darker basophilic nucleus with small amount of cytoplasm right here. Small amount of cytoplasm, which is basophilic once again. But these cells are not so active in terms of production. So just remember, inactive state of fibroblast is called fibrocyte. Yeah. Here you can see also the difference between these two. Here we can see a picture from electron microscope. So once again, really narrow cell right here, narrow cell, spindle shape, but we cannot see it right here because it's just a part of a fibroblast, but very light oval nucleus right here. So there is the nucleus, and you can see also granular endoplasmic reticulum, the cisternae of granular endoplasmic reticulum right here. So that's why the cytoplasm is basophilic. And it also contains a Golgi complex. And here we can see vesicles right here. So probably these vesicles, we can't really tell if they are spinocytic vesicles or if they are these vesicles which contain procollagen. So we can't really tell that, but probably they are this. No, that's not what I meant. Really, these are just vesicles, and we can't tell if they are pinocytic, or they need to be excreted because they contain procollagen. So that doesn't matter right now. But we need to know that fibroblast isn't storing collagen inside the cytoplasm. It's constitutively released outside. And remember that really after that, when a tropocollagen is released, tropocollagen doesn't contain registration peptides, so it can polymerize tropocollagen, and when tropocollagen polymerized, it creates first protofibrils, and after that it aggregates into fibrils. And please remember collagen synthesis. It's very important to know collagen synthesis during oral exams and also for the tests. So you should know it. What's happening really in the fibroblast and what's happening outside of the fibroblast. What we can see in electron microscope, we can see fibrils right here, and together these fibrils, which are pink here in my picture, these fibrils create a fiber. This is a whole collagen fiber, and this fiber right here can be seen in light microscopy. And this is very important to differentiate fibrils in electron microscopy and fibers in light microscopy. So don't mix these terms, please. It's important. So this is a picture of fibroblast. And here we have a myofibroblast here in this picture. It's not so important to differentiate myofibroblast in electron microscopy or in light microscopy in general, but you should know that they exist and they lack the superficial lamina, and they contain structures that can be found also in muscle tissue. So here we have written the dense bodies. You're going to learn more about them during the lecture of muscle tissue, but you just need to know right now they're able to contract, because they contain structures that are in the muscle tissue. So these myofibroblasts also sometimes can be found in proper connective tissue. And then we have a reticular cell. Reticular cell, a star-shaped cell with lot of processes. These reticular cells create stroma of many organs. So they create stroma. like a skeletal structure that holds parenchyma together, or cells together. As I mentioned before, the spleen, the lymph node. So we can find reticular cells there, and also these reticular cells are specialized for the collagen type 3 production. So collagen type 3. Yeah, so here we have nice reticular cell right here, and here we can see many collagen fibrils right here, many collagen fibrils, which together create a reticular fiber right here. And they create this network. Also the fibers, reticular fibers, together with these cells, create a network in the organs, the stroma. Next, cells that can be found in proper connective tissue, and also the cells that create one type of connective tissue, or one type of the proper connective tissue, the adipose tissue, are adipocytes or fat cells. Adipocytes are a little bit deviated from the general rules of connective tissue, because these adipocytes alone have the ability to store fat. Yeah, so that's why they are fat cells, adipocytes. And they grow, they are very big, and those fat droplets, these fat droplets, remember these are the inclusions in the cell, these fat droplets often merge together. Yeah, so adipocytes are formed from a mesenchymal cell, undifferentiated cell of a proper connective tissue. These mesenchymal cells can differentiate into fibroblasts, but also they can differentiate into lipoblasts. So lipoblasts is undifferentiated cell of adipose tissue, and these lipoblasts can create either unilocular adipocyte, which have one big fat inclusion inside, because these lipid droplets merge together and fuse together and create one big lipid droplet inside. But they are also smaller ones in the cytoplasm here, but we can see often this one big droplet right here. Or they can create something called multilocular adipocyte, multilocular, which has these many lipid droplets inside the cytoplasm, and it's very typical for the brown adipose tissue. These cells create brown adipose tissue, and these cells create just normal adipose tissue in the human body. Yes, but remember this lipoblast, undifferentiated cell. Yeah, right here. It's very similar to multilocular adipocyte. Yeah, sometimes you cannot really differentiate. This is a lipoblast or it's multilocular adipocyte. Yeah? So sometimes it's hard to tell. Yeah, so here we have a picture from light microscope. On one hand, we can see unilocular or univacuolar adipocytes right here. So this is normal adipose tissue. And what we can see right here are these big lipid droplets. So many small droplets fused together, and they create these big lipid droplets lipids were washed out. So there's empty space right here. So there's nothing right now in here. And what we can see around the lipid droplets, this pink line right here, what we can see around them, this is the cytoplasm. Really, this is the cytoplasm of adipocyte around here. And in this small amount of cytoplasm around the lipid droplets, here we can find all organelles of adipocyte. Here we can find also nucleus. Yeah, also nucleus. So, for example, I don't see quite good cell, but for example, here we have a cell, adipocyte right here, and this basophilic dot that is right here, this is probably a nucleus of an adipocyte, of an unilocular adipocyte, and it's really on the periphery, because there is such a big lipid droplet in the center, so everything is on the periphery, all the organelles, and also this nucleus is located on the periphery. So this is what you can see right here. Yeah. And when you look very closely on the other side, we really have multilocular or multivacular adipocytes right here. So once again, this is a really not so clear picture. So really look, for example, at this adipocyte right here. And what we can see here, we can see many lipid droplets. Here, right here, and what we can see in the center, here we can see the nucleus right here. Yeah, so, and for example, next thing, right here is another cell, and look very closely, you can see some droplets here. There are many of them, they are not creating this one big lipid droplet. And here we have a nucleus. It's not quite in the center, it doesn't have to be, but it's not on the periphery of the cell. It's not on the periphery. So that's why. And sometimes really unilocular or univacular adipocytes, like here, for example, yeah, when you look very closely, you can see this nucleus on the periphery. We don't really can tell, like, these are free cells right here, 1, 2, 3, and we can't really tell, like, is this nucleus, does this nucleus belongs to the cell number 2 or 3? It's hard to tell, but sometimes we can see adipocytes which, like this one, which doesn't even have nucleus. And it's not that this adipocyte doesn't contain nucleus. It's just this situation that adipocyte is so big, yeah, with the nucleus. This adipocyte is so big that it was sections somewhere else than through the nucleus. Yeah, so that's why we can't see nucleus on every adipocyte here. These multilocular adipocytes, as you remember maybe from the cytology, they contain protein called thermogenin protein. It belongs to a family of uncoupling proteins, and they sit next to ATP synthase in mitochondria. So in mitochondria, you know that mitochondria produce ATP, and here we have this ATP synthase, and thermogenin is a protein that sits next to ATP synthase, and it's uncoupling this ATP synthase. That means that the protons, the flux of the protons is used to produce heat, not energy in this case. Yeah, so ATP synthase isn't working properly. Sometimes it's producing ATP also because these cells needs to have ATP because they need energy. Every cell needs energy, but they also have this thermogenin, so they produce also heat. Yeah, not only ATP. And as I told you before, these adipocytes have something called external or superficial lamina on the surface, so also muscle tissue, but also these adipocytes have external or superficial lamina. Yeah, it's written here in this picture. That is a basal lamina right here is written right here is basal lamina BL, but it's not really basal lamina because adipocytes are not polarized. So we cannot call it the base of an adipocyte. We call it in our department, it's a superficial or external lamina because it's around the cell, not only in base of the cell. Yeah, but this electron micrograph isn't that important, really. You can see a big lipid droplet and a small amount of cytoplasm. Yeah, these are two cells that are close together, and really adipocytes have only a small amount of tissue between them. Extracellular matrix. Yeah, only small amount of extracellular matrix between them. So really, adipose tissue is a little bit different than a normal connective tissue, proper connective tissue. Yeah, and here we have a picture of a macrophage. So let's talk about migratory cells. Yeah, for now on. Here we have a picture of macrophage. This is a picture from electron microscope. Of course, macrophages are wandering or migratory cells, and the macrophages are specialized for phagocytosis, so they eat. Yeah. Macrophage measures about 30 micrometers, so it's quite big. 30 micrometer. We can't see macrophages in a light microscope, of course. They are so big in electron microscope. And macrophages have a lobed, or they have some kind of indentations in the nucleus. It's not very good here, but what you will see in light microscopy, they look like this in light microscopy. And they have a nucleus that is like splitted into two parts like this. This is typical nucleus for a macrophage. And this part right here, this invagination in the nucleus is called indentation. Yeah, so I'm going to write it down to be sure that you know this term, indentation. And it's very typical for macrophages. They have eosinophilic cytoplasm, very eosinophilic cytoplasm, basophilic nucleus, of course. Nucleus is always basophilic, with indentation or invagination of a nucleus. Yeah, and macrophages contain a lot of lysosomes, and these lysosomes are in different stages of development. Right here. We have lysosome right here, and different stages are, for example, we can see lysosomal vesicles. Yeah, so these, for example, are the lysosomal vesicles right here, small, and these are bigger lysosomes right here, early lysosome and some kind of late lysosomes, which are digesting right here. And here we have very late lysosome that is almost empty because it digested everything, almost everything. So here we have lysosomal transport vesicles, which contain enzymes which are not active yet. But when these lysosomal transport vesicles fused with late endosomes, which contain this digested material, they create another stage of lysosomal system, and that is a lysosome. So lysosome is the biggest structure right here, and this is early lysosome, which has active enzymes, and here we can see late lysosome, which is somehow like digesting things, so its structure looks very odd and different. And also here we see this digestion right here, vortex is inside and something's happening here, and also here. And this lysosome is kind of empty, so probably this is very late stage, probably a terminal lysosome which doesn't contain enzymes because everything was digested already. So yeah, this is a macrophage. And also you can see its structure on the outside. Yeah, and here we have this pseudopodia. Pseudopodia are like little legs because macrophages can move actively. Yeah, so they can move from the they were created from monocytes, which are immune cells that are in the bloodstream. And when these cells are attached to the vessels and they migrate to the proper connective tissue under vessels, and when they do so, they differentiate into macrophages. Yeah, so, and these are cells specialized for digestion and phagocytosis. They're able to work for several years. Yes, so they don't eventually die right after. They work for several years. Yeah, so, but also where we can find them, so we can find them in proper connective tissue. And there is this picture from the lungs. These are the lungs. You would be able to recognize lungs in the next semester. But these are the alveoli in the lungs. So these spaces right here, these are the alveoli. And what we can see right here, we can see these guys. And these are macrophages that are located in the lungs. Yeah, so these are the macrophages that are located in the lungs, or alveolar macrophages. And you can see that they are kind of brown. Yeah, they are brown because they digested dust. Yeah, in the lungs, they digested the dust. So what we can see really inside these macrophages, these are the dust particles, and that's why they are so brown in here. And when they do so, when the macrophage digests or phagocytose dust, and they are located in the alveoli of the lungs, they are called coniophages. Coniophages. So coniophages are macrophages that digested dust in the lungs. So it's just the same cell. It's the same cell as macrophage, but with the dust inside. Yummy. So yeah, these are the coniophages. And we have macrophages also in the spleen when they are digesting red blood cells in the spleen. And when they do so, these particles of red blood cells can be seen also in the cytoplasm of macrophages. And when macrophage digests a red blood cell, yeah, this is a picture of red blood cell, I'm sorry. But when they do so, when they digest these red blood cells, they are called siderophages. Siderophages. Yeah, so these are just two types of macrophages and depends on what structures they have eaten, really. Okay, the next is the mast cell. Mast cell is a very special type of a migratory cell that can be found in proper connective tissue. Here we have a picture from electron microscope and also from a light microscope. You can see that mast cells contain large granules on the inside. They are spherical shaped, and also they have these spherical shaped granules on the inside. They are very electron dense in the light microscope, very electron dense granules here. But they are basophilic in light microscopy, basophilic, and they have this phenomenon while staining. It's called metachromasia. I'm going to write it down. Metachromasia. It's typical for these granules of a mast cell, and this generally means that I'm going to make an example. Like, for example, we stained this mast cell right here with toluidine blue. Toluidine blue. So these granules should be blue, yeah? But they change color when stained. They are violet. And this is the metachromasia. It's a really phenomenon which means that the color of the dye we used changes when it stains the structure. So it should be blue, but it changes when it stains the structure. So now we can see these granules are violet and sometimes even more purple or reddish, yes, but we can see violet granules right here. Many granules that are here closely packed together. And this is a metachromasia. It's typical for the mast cell. I don't think it's that important, but I should tell you that. Sometimes you can see it written in textbooks, so that's why I'm telling you this. So once again, it's a phenomenon, which means that the color of the dye or of the staining changes when it stains the structure. So we have this blue color, but when it stains the structure, it's violet. Yeah, so that's the metachromasia. It's like magic. Not so much, but yeah. Really, the mast cells produce low molecular weight substances such as histamine or heparin or chondroitin sulfate. So which are very important. The most important here are histamine. You should remember that, and heparin. And you know these two molecules. You know why do we have histamine and heparin for sure. These are important mediators of inflammation. So histamine is dilating the vessels. Yeah, so when you have too much histamine, you're red. The skin is red. It's for the inflammation, because you need to dilate vessels for the inflammatory cells to come and destroy something that is triggering the tissue, that some kind of antigen or something that's there. So histamine is for the vasodilation. Yeah. And heparin, you probably know as a main anticoagulant. Yeah, but it also is important in the process of inflammation. Yeah, so these two molecules are inside these granules. So this is the mast cells. These are the mast cells. And this is a picture from electron microscope, and we can see these granules right here. These are the granules. So sometimes we can see them as electron dense, but here we can see that these granules sometimes contain these vortexes, which are typical for mast cell granules, but this is not so important. And next we have a plasma cell, which you already know. Plasma cell is an effector B cell, B lymphocyte, which these plasma cells produce immunoglobulins. So once again, they produce antibodies, and you know that these antibodies are stored in the granular endoplasmic reticulum. So granular endoplasmic reticulum is dilated because these plasma cells are storing the immunoglobulins. They are waiting for the signal to release immunoglobulins, and the signal, which is important here, is an antigen. So when they meet with antigen, they release immunoglobulins from the granular endoplasmic reticulum. They have very typical nucleus, which is called clock phase nucleus. So heterochromatin is located peripherally and creates these spikes right here. Yeah, so it's a clock phase nucleus, very typical. I hope we have a picture from light microscope. Yeah, this is a picture of a light microscope from a connective tissue, loose connective tissue, and we can see some fibroblasts right here, active cells, and here we have a plasma cells. So there is this one plasma cell over here, and there is a second plasma cell over here, and plasma cells are more triangular shaped, with basophilic cytoplasm, of course, because it contains granular endoplasmic reticulum, and they have this clock phase nucleus in some point of the triangle right here, clock phase nucleus like this. So these big cells right here are typical plasma cells. Yep, so, and here we have a lymphocyte, of course. Oops, sorry. And collagen is all around this pinky. Extracellular matrix is collagen, yeah, collagen fibers. These are the fibroblasts, and also we can find lymphocytes as immune cells here. There could be also a granulocytes and other immune cells that you're going to learn about more. And this is a picture from electron microscope right here. This is the plasma cell, and we can see the nucleus right here. Nucleus. And here we can see dilated cisternae of granular endoplasmic reticulum right here. Dilated cisternaes which contain immunoglobulins right here. So this is a very typical picture for the plasma cell. Well, and we have many types of proper connective tissue. And this is a very important slide. You should know and you should describe all types of proper connective tissue. So we can find something called mesenchyme, yeah, and mesenchyme is an embryonic tissue which contains mesenchymal cells and a small amount of extracellular matrix. Yeah, so mesenchyme creates embryonic tissue. It's a very undifferentiated tissue. Then we have a mucous connective tissue which creates this umbilical cord, or is sometimes called Wharton's jelly. Mucous tissue can be found also in dental pulp, but only in kids or small children, because a dental pulp in adults is more differentiated. You can find more fibers in there and also collagen fibers. So only dental pulp of kids is made of mucous connective tissue. We can find fibroblasts there, and we can find hyaluronic acid there in large amount. So large amount of hyaluronic acid in the mucous connective tissue, and it's typical for the reticular fibers also. So it's like that. These are very similar. The mesenchyme and the mucous tissue are similar. But embryo itself, when it reaches that stage of the fetus, you know that we have an embryo for the first 8 weeks, and then we have a fetus after 8 weeks. So from 9 weeks till the labor, we have this fetus. And the mesenchymal tissue changes to the mucous connective tissue. So we can find mostly mesenchyme in the embryo, but it changes to a mucous connective tissue in the fetus after the 8th week of development. Yeah. So just remember, mucous connective tissue, we can find it in the umbilical cord. That's very important because we have one slide or 2 slides with umbilical cord, cross section of umbilical cord. That's typical for mucous connective tissue. So you should describe mucous connective tissue. Yeah. And we can find it also in dental pulps of children. We can find their fibroblasts, so no mesenchymal cells are, but differentiated fibroblasts are here in mucous connective tissue. Large amount of hyaluronic acid, that's very typical, and reticular fibers. Yeah, so really collagen, next is collagen connective tissue. And that's the most common. It contains all components. Yeah, so most common is a collagen connective tissue. It can be divided into loose connective tissue and also dense connective tissue. And you should know differences between these two. Loose connective tissue, loose collagen connective tissue contains thin fibers of collagen, also of collagen type 1, 2, and 3. Not 2, I'm sorry, because you should know that collagen type 2 doesn't form fibers. So we can find only type 1 collagen fibers, type 3 collagen fibers, and also elastic fibers. Yeah, And we can find more cells in loose connective tissue, like fibroblasts and many migratory cells. On the other hand, dense connective tissue contains thick fibers, thick collagen fibers. There aren't more of them. You cannot say that there are more fibers in dense connective tissue. They are just thicker collagen fibers, and dense connective tissue contains less cells, yeah, and mostly fibrosites, which aren't that active. Yeah, fibrosites are inactive form of fibroblasts. Dense connective tissue can be divided into regular connective tissue, which is in the tendons or ligaments. We're going to have a look on regular connective tissue, or irregular. Irregular dense connective tissue can be found in capsules that line organs, many capsules. So each capsule that is around organ is made of dense irregular connective tissue. And for example, in the dermis, in the skin, we can find this dense irregular connective tissue. It's important to say the whole name, because when you say just dense connective tissue, it's not very correct. You should all the time during histology, you should say it's dense irregular connective tissue, yeah, or loose connective tissue. Really, what's happening in the human body, sometimes you can't tell the difference between dense and loose. They are these transition zones where we are not entirely sure what kind of tissue it is, whether it's like loose or dense. So sometimes you can't really tell. Yeah, but most of the time you will be able to tell this is loose connective and this is dense connective tissue. Yeah. And really, this dense collagen connective tissue, when it's regular, that means that these fibers are parallel to each other. Yeah, so they are not just like mixing in some network, but they are parallelly arranged to one and another, and they create this bigger structure, the tendon. Yeah, you probably cannot imagine tendon right now, but you will be able to do that because tendon is this very like arranged structure. It's not just like some kind of heterogeneous structure. It's very homogeneous, and fibers in the tendon are parallelly arranged closely together. Then we have this, so we covered mesenchyme, mucus, collagen, and then we have this reticular connective tissue, which can be found in lymphoid organs and also in the bone marrow. And that's why this reticular connective tissue is creating a stroma for the lymphoid organs. And for example, in lymph node, we have this stroma that is created with reticular connective tissue. Imagine it like reticular cells with reticular fibers. That's the reticular connective tissue. And the parenchyme of a lymph node is created via lymphocytes or immune cells in there. Yeah. So reticular connective tissue is just stroma. And you already know reticular cells. These are cells which are specialized for the production of collagen type 3. So they create only reticular fibers, which are thinner than collagen fibers and not so well arranged. And they are highly glycosylated. Then we have elastic collagen tissue. Elastic connective tissue. Elastic connective tissue creates yellow ligaments of the vertebral column, the yellow ligaments. These yellow ligaments are between these structures that I forgot the name of these structures that create vertebra that are between the vertebra. I'm sorry. So we can find elastic connective tissue there. This connective tissue, it's made of fibrocytes and elastic fibers. Yeah, of course. And then we have adipose connective tissue, which is made of adipocytes. And we can find brown adipose connective tissue, which is made of multivacuolar adipocytes. You remember that, producing heat because of thermogenin that can be found in kids, or normal adipose tissue that can be found in everybody. Yeah, everybody has adipose tissue, normal adipose tissue. Okay, let's have a look on these types of tissue. That was really difficult right now. You should be able to divide the proper connective tissue into these types. So please remember the basics right here from this slide, and then let's have a look. Once again, mesenchyme tissue, very undifferentiated tissue right here. These cells are very light, they're very active, there is just a small amount of extracellular matrix. You probably wouldn't be able to see this type of tissue, but what you can find on slides is this mucous connective tissue. Yeah, this is the mucous connective tissue right here. So once again, these are the fibroblasts and fibrocytes right here. We can't tell sometimes, but okay, let's call them fibroblasts right here, these dark cells. Yeah, and you can see this large volume of extracellular matrix. So we have this large volume here. Among these fibroblasts, we can see finely eosinophilic fibers. So we can see these fibers right here. And the intracellular mass is mostly amorphous. These are the empty spaces. Yeah, empty spaces right here. But here we can find really the molecules, the amorphous component, yeah, of the extracellular matrix. So here we have these fibers, this is the fibrillar component, and these empty spaces contain amorphous component. This is hematoxylin eosin staining, but we have one staining that Alcian blue that you can find also in our slides. So we have umbilical cord that is stained with hematoxylin eosin, and we have umbilical cord stained with Alcian blue. And this Alcian blue, we will really see blue-colored umbilical cord because it stains hyaluronic acid, mostly hyaluronic acid, in the amorphous component of extracellular matrix. So these spaces which are here kind of look like empty, like it's nothing in there, they are very poorly stained. They will be blue with Alcian blue. Yeah, so just remember Alcian blue for the umbilical cord. We are staining umbilicals with Alcian blue because we want to see amorphous component there, and this hyaluronic acid is very well stained with this blue color. Yeah, but not in this slide. This is normal, just normal hematoxylin eosin. Next we have a picture, very nice picture from loose connective tissue. So this is very typical loose connective tissue. We can see fibrocytes right here. These are the fibroblasts. This is the fibroblast, very active nucleus. This is the fibrocyte, darker, smaller oval nucleus right here. Yeah, fibroblast right here, and here is fibrocyte. Yeah, but there are many of them here, as you can see. We can see collagen fibers right here. So these eosinophilic structures right here are collagen fibers, but probably also reticular fibers and also elastic fibers, but elastic fibers aren't stained that well as collagen fibers. So maybe some fibers that are poorly stained here are elastic fibers, but we can really tell for sure. We don't have special staining here, just hematoxylin eosin, so we can't tell for sure. Here we can see some collagen fibers are thinner, some of them are thicker, like this fiber right here, so it differs. So fibroblast, and here we can see also some migratory cells as plasma cells. So here is nice plasma cell over here, and also here is the plasma cell with highly basophilic cytoplasm. Also fibroblasts have basophilic cytoplasm. And here, for example, there is a nice little, no, it's not that little, but here we have a macrophage. Yeah, macrophage right here, and we can also see dust particles or some particles in here. Maybe this isn't dust because we're not in the lungs, but we can see some particles, some garbage from the world that is around him. So probably is cleaning something here. Yeah, so we can see macrophage right here. Yeah, this guy. And the loose connective tissue is highly vascularized. So here we have vessels. These are the vessels. Some arterioles and venules. So here we can see vessels right here. And you can see it's not organized. You have cells there, migratory fixed there all around. Here vessels are everywhere, and fibers are, some of fibers are going one way, other fibers are going other way, so it's not organized at all. And there are thin collagen fibers here, very thin collagen fibers. Then we have dense irregular connective tissue. And dense irregular connective tissue contains thicker collagen fibers. So these eosinophilic structures right here, yeah, this is a collagen fiber, this big structure here. Remember, we can't see fibrils, we only can see fibers in light microscopy. So these are the fibers. And this is a longitudinal section of a fiber, and this is a cross section of a fiber. And that's why we can tell this is dense irregular connective tissue. Because we can see these longitudinal sections and cross sections. Yeah, that's not arranged at all. These fibers are going different ways. So that's why it's irregular connective tissue. And this is typical, this connective tissue is typical for the dermis, for example, and also for the capsules, which are surrounding organs. Yeah. So dense irregular connective tissue. Well, maybe I can show you one thing. Yeah, that, okay, so we have thicker collagen fibers, but also we have some cells here and here. These are probably the fibroblasts and fibrocytes here. And there is only small amount of cells here in dense connective tissue. Yeah, compare it to the loose connective tissue. Many cells, thin collagen fibers, and here less cells and thicker collagen fibers. And it's irregular because we can see longitudinal and cross sections at the same time here in this photo. And here we have dense regular connective tissue. Dense regular connective tissue, which is typical for the ligaments and tendons. Yeah, that's the same thing, I guess, but you can find them in tendons. And what we can see right here, we can see these collagen fibers that are very thick collagen fibers right here. And between these collagen fibers, we can see these narrow cells that are fibrocytes, probably. Fibrocytes. So this is a longitudinal section of some tendon, I guess. And this is a cross section. Cross section So we can see cross sections of collagen fibers right here, yeah, which is highly eosinophilic. But all fibers are going to the same direction. All of these are cross-sectioned and these are longitudinally sectioned. So that's why we can tell this is dense because of the thickness of these fibers, very dense and small amount of cells, regular connective tissue because all fibers are going in the same direction somewhere. So this is a picture of tendon. Once again, it's not stained very well. It should be eosinophilic, more eosinophilic, but we can see these collagen fibers going for the one direction. This is one collagen fiber right here. It's very thick. So this is once again a dense, regular connective tissue. And we can see fiber sites that are between these collagen fibers right here. Next, we have reticular connective tissue, and I'm sorry for this letter here, it should be red, but that does. So reticular connective tissue right here, and this is the picture from the spleen, for example. So there is not only reticular connective tissue here, which is made of reticular cells and reticular fibers, but we can also see many cells here, which are the cells of an immune system. But what is really a reticular connective tissue? This is once again an impregnation, because normally we wouldn't be able to see reticular fibers that well. And reticular cells also. But in impregnation we can see these black lines right here, and these are reticular fibers right here. Reticular fibers. And we're going to show you reticular cells in the next semester, when we are going to describe the whole structure of the spleen and lymph nodes and stuff like this, because it's sometimes hard to differentiate them in such big magnification, that in such a small magnification that we can see right here. But just now, like, look at this reticular network. These are reticular fibers. They are black because of the impregnation of the salts and with the salts, and sometimes these fibers surround vessels like this, so you can find many of them around vessels. Here is the vessel. I know it's really hard for you to tell where are the vessels now, but don't worry, we cover that later. So just remember this picture, the black lines of reticular fibers, which create reticular connective tissue together with reticular cells. Next we have elastic connective tissue. This is a picture from light microscope both. This is hematoxylin eosin staining, and you can see it's pink, but it's not stained very well once again. So here we have these elastic fibers right here. They're kind of thick, but they aren't stained very well. And do you remember they're made of two components, the microfibrils and also this elastic core in the middle. So you should know that. And yeah, they are not very good stained with hematoxylin eosin, but sometimes we can find these elastic fibers also in the vessels. Do you remember the vessels? And they are wavy here. So this is some kind of structure, a ligament that was made only of the elastic fibers. So this is really example of elastic connective tissue right here. But we can find elastic fibers by themselves in many structures. We can find elastic fibers in loose connective tissue. We can find elastic fibers in the walls of the vessel, like these here. They create these fenestrated membranes right here. And this is a special staining because you can see they are brown right here. And another special staining for elastic fibers is orsein. Yeah, so this is another special staining for elastic fibers. so you can really see them, because without that you will not be able to see them that well, that good. So once again, this is white adipose tissue. You can see many cells. This is white adipose tissue because it's made of unilocular cells. So these guys are here. We can see these big lipid droplets and small amount of extracellular matrix. So adipose tissue is not regular connective tissue, proper connective tissue, because there is just small amount of extracellular matrix. Yeah, so it's not that typical connective tissue. And we can see vessels in this extracellular matrix. So once again, remember, adipose tissue is kind of different. White adipose tissue is made of unilocular or univacular adipocytes, these big guys right here, and we can see small amount of extracellular matrix around these adipocytes that contain vessels, like here. And the last picture of my presentation will be brown adipose tissue once again, and we can see the localization. So it's suprascapulary around the scapule in the back region of the kid, and somewhere here in the abdominal region right here. So brown adipose tissue is specialized for the heat production, and it's made of multilocular adipocytes right here. So we can see them right here. Sometimes you cannot really differentiate the borders between these cells, but what you can see here, you can see these lipid droplets right here. Yeah, so that's what you can see, and that's how you can differentiate. And don't forget about thermogenin. Yeah? Thermogenin and uncoupling protein, which is located. And why is it called brown? Brown adipose tissue is really called brown because these cells contain a lot of mitochondria. And cytochrome in this mitochondria has a natural brown color. So that's why it's a brown fat or brown adipose tissue, because of the cytochrome inside the mitochondria. Yeah, so yeah, and you can find it in newborns, a minimum amount in adults. Yeah, okay, so this was the last slide, I guess. So thank you and have a nice day, and I hope you enjoyed this lecture. But if you struggle with something, please tell me, write in the comments or send me a message or whatever you want to do. And okay, so bye bye, thank you.